Okay, today we will learn about mitochondria. Mitochondria. यदि हामीहरूले कुनै सेल लियो भने त्यो सेल त एकदम it is too small to see, and if we see it by a compound microscope also, then we will only see the outer contents, cell wall, cell membrane, and the cytoplasm present inside it. We can't be able to see the cell organelles present out here. So if we will be able to see, then also then how we will know that which cell organelles we are knowing. So to see mitochondria, we use a stain, a special type of stain known as Janus green. Janus green. It is a stain. Stain means a substance, chemical substance, which is used to color a specific type of substance. This Janus green is used to color the mitochondria. So when we put a Janus green in it, it colors out the mitochondria, and we are easily able to see the mitochondria by putting this Janus green. Now if you will look at the shape of the mitochondria, then it will be looking like something elliptical. It will be like something elliptical to be sausage shaped. हम लोग whenever we used to see in cartoons, and then there we see a sauce. Sauce is what? It is a meat packed in a different shaped, special shaped, and that meat is packed in something like this shaped. Okay, this shaped is the shape of sauce, and mitochondrial shape resembles with the shape of the sauce. So mitochondria is termed as sauce shaped. And if you look at the morphology, then if we will find the diameter of mitochondria will be 0.2 micrometer to 1 micrometer, and length will be equal to 1 micrometer to 4.1 micrometer. Diameter is 0.2 to 1 micrometer, and length is 1 micrometer to 4.1 micrometer. Now this variation in diameter or dimensions in diameter and length is due to the physiology of the cell. Means if this mitochondria is present in such type of cell where the energy requirement is more than it of course it should have more it should have to be more large to provide that or to sustain that energy demand. So depending upon the physiology of the cell, this mitochondria changes or varies from its dimensions. If we look like the ultra structure of mitochondria, then it is a double membranous cell organelle, and wherever you find double membranous cell membrane, and then you should keep in mind that outer membrane is permeable and inner membrane is selectively permeable. Permeable means it allows all the substances, all the molecules to pass through it, and selectively permeable means it allows only some special types of substance, like it allows only glucose molecules, pyruvate molecules to pass through it. Then it has selected, the membrane has selected only certain compounds to pass through it, then it is called selective permeable. So this is inner membrane and this is outer membrane. Sorry, this is inner membrane, this is outer membrane. So outer membrane is selective and permeable, and inner membrane is selectively permeable. And if we will see about the microscopic zoom of mitochondria, then we will find if this is outer membrane and this is inner membrane, then in the inner membrane we will find some the functional unit of mitochondria, that is oxysomes, or this is called oxysomes or F1 particles. So these mainly plays roles in ATP synthesis. What it does that if this is outside of the cell body, and during some of the chemicals going chemical reactions going out in the cell, protons are produced, and these proteins concentration outside the mitochondria is too high, okay, and inside the mitochondria the concentration of H plus protons is too low. So due to the variation in potential gradient of protons, potential gradient is nothing matlab if you will divide if there is a boundary, and if we take anything water or anything people's population, and then if here 200 peoples are present and here only 10 peoples are present, then this difference in density or this difference in number of peoples is stated to be population gradient, means difference of population with respect to length. यहाँ बॉडी पपुलेसन छ, यहाँ कम पपुलेसन छ, त्यस्तै प्रोटोन्स ग्रेडियंट means outside the mitochondria the protons level is high, and inside the mitochondria the protons level is low. So what it does that it just allows the protons, due to the potential gradient, the protons move inside the mitochondria, and if due to the potential gradient it moves, and while a body is moving, then we know that if a body is moving it carries kinetic energy, and that kinetic energy is utilized by these oxysomes or F1 particles to convert ADP into ATP. Now I will just say what is ADP and ATP. ADP is nothing, just molecules that has 2 phosphate ions, and ATP means molecules that have 3 phosphate ions. There is a detailed structures that we don't have to study, but we know that. Let me just tell about the what is the main difference between ADP and ATP by describing its structure. ADP and ATP are just nothing. They are glucose attached with adenine. Adenine is a double ring structure, hexagonal 1 2 3 4 5 6, and here NH2 is attached, and phosphate ion minus PO4 minus. If 2 phosphate ions are attached, then it is called ADP, and due to the kinetic energy produced by the proton, that energy is utilized to make a bond between ADP, these 2 phosphate groups, and due to which it contains, as earlier it was containing 2 phosphate ions, it was stated to be adenosine diphosphate. Di means 2 phosphates. Now due to the connection of one more phosphate group, it is stated to be ATP, adenosine triphosphate, means 3 phosphate group connecting to it. And how we get the energy from ADP and ATP? It just normally when a body needs energy, the bond between this phosphate breaks down, and that the energy released out due to the breaking of that bond, that is called phosphodiester bond, and breaks out, and due to which the energy is released, and that energy is utilized by our body in doing many various functions.